Hello guys, welcome to IB360. We will discuss today about mitochondria. Mitochondria is a very special cell organ, as you all know, and the word mitochondria, this was actually discovered, or rather the term mitochondria coined by Benda in 1897. Back in 1880 it was first observed by Colliker, and behind the naming of mitochondria, the words mitose and chondrion played roles. Mitose means thread and chondrion means grain. So Benda in 1897 termed the organ mitochondria. Mitochondria can be easily stained with genus green and observed under light microscope, genus green stain, but its ultrastructure can only be observed under electron microscope. Okay. As far as functions are concerned, mainly two important physiological processes are happening, or we can say cellular processes actually. One is oxidative phosphorylation, and second is Krebs cycle, which is the second stage in aerobic respiration. When you study respiration in case of plants or animals, you'll come across both of these mechanisms or phenomena, and both of them occurring inside mitochondria. Occurrence, see, mitochondria, obviously they would be absent in prokaryotes as well as anaerobic eukaryotes, but other than these basic facts, some interesting information I can give you about occurrence. So let us write down the points one by one. The first thing is absent in prokaryotes and anaerobic eukaryotes, because if they are anaerobic, then obviously they will not undergo Krebs cycle. Everything about respiration will take place inside cytoplasm itself, and also in prokaryotes they have the mesosomes, which are special cytoplasmic invaginations, or rather plasma membrane invaginations into the cytoplasm, which are kind of replacing the roles of Golgi apparatus or mitochondria as such. Second is they are also absent in mature RBCs. Why they are absent in mature RBCs? Because RBCs, which are the oxygen, which are having hemoglobin, which is the oxygen transporter in blood, if RBCs have mitochondria, then the oxygen which they carry, that would be used up in cellular respiration in itself. So before transporting the thing to the required tissues, it is using it up itself. So that can't be afforded. So mature RBCs, they're losing their mitochondria. In fact, they don't have endoplasmic reticulum to maintain flexibility. There is another issue, but this is the thing. Third point is that in case of cells which are not undergoing rapid respiration or don't have that much amount of energy requirement, for example cells of dormant seed, they will have fewer mitochondria, not much, because there is not much of a requirement on so in stark contrast to this, we have cells of germinating seed. They have abundance of mitochondria. Okay, another important aspect of occurrence is, so first of all you will understand that this availability of mitochondria, highly situational factor. Second is their positioning. So in unspecialized cells, they are randomly distributed. in the cytoplasm, but in case of absorptive cells or secretive cells, they would be found more towards the peripheral cytoplasm, more towards peripheral cytoplasm. Even during cell division, during cell division, they would be found more towards the spindle fibers, because these are the locations which require energy. So according to the situation, according to the processes happening inside the cells, or according to the type of cells, their occurrence, their positioning will vary. Okay, now we will talk about the ultrastructure of mitochondria. We have already discussed its primary functions, occurrence, and the discoverers and observers. So mitochondrial ultrastructure can be observed under electron microscope, not under light microscope. In this case, staining with Janus green will not work. We have to have an electron microscope to observe all these things. Now, the most prominent thing when you look at a mitochondrion is you will observe these two membranes, the outer membrane and the inner membrane. So these two membranes are dividing the entire mitochondrial space into two chambers. So in this diagram you see a transverse slice. Obviously it will have another half. They have kind of cut through it like this. It's a transverse section of mitochondrion. Here in this diagram you see there are the two chambers clearly can be seen, the outer chamber and the inner chamber. The outer chamber is also called the perimitochondrial space. Inner chamber is filled with semi-fluid substance which is called the matrix, the ground substance. The outer membrane compared to inner membrane has some differences which it shows. So the outer membrane is relatively smooth and also permeable to a large number of metabolites, so more permeable. This is less smooth and permeable only to certain metabolites, so selectively permeable, selectively or less permeable. Okay, then you see there are ribosomes in the inner chamber, DNA. So these presence of these elements support the fact that mitochondria shows autonomy. It has its own ribosomes which are usually 70S types or sometimes 55S types. It has its own DNA which is circular and naked, and also it has RNAs which are produced by its own DNA. Now, so here also only we can mention that DNA is circular and naked. These two important things are there. And ribosome can be 55S type or 70S type. Okay, and you see that there are certain invaginations. These are the invaginations or folds of the inner membrane itself. Now, as the inner membrane is folding on itself in that fashion, some of the outer chamber is protruding into the inner chamber, and these are known as cristae. So see, this is the cristae we are talking about. Now they are very vital and they are very significant in certain respects. At the very first slide we had talked about two primary functions that the mitochondria undergo. One is Krebs cycle and second is oxidative phosphorylation. You must remember this for the moment, that Krebs cycle or TCA cycle takes place in the inner chamber. Enzymes which are required for Krebs cycle except one are found inside inner chamber. And the electron transport chain, which is the final process involved in aerobic respiration, through which ultimately the NADH rather is converted to ATP, that takes place in the inner membrane. Okay, so one process is happening in the inner chamber and one process is happening in the inner membrane. And here, as the cristae are existing, they are increasing the surface area for the inner membrane, and this is how they are helping in an increased amount of the ETS process, the electron transport system process. More and more that is taking place, and more ATP can be generated if required by the cell, because obviously mitochondria is known as the powerhouse of cell. We have talked about this since our childhood. Okay, so we will talk about cristae in much more details actually, but before that, another important point of difference between outer membrane and inner membrane, actually these three are enough. The third point being the availability of proteins. So outer membrane is poor in proteins, but inner membrane has a lot of proteins, like 70 to 75% of total components, they are proteins. And another important thing which you will see in the next diagram is the presence of porins in the outer membrane. Porins, these are pores, minute pores, and they are actually protein channels. Obviously they are facilitating entry of substances into the outer chamber from the cytoplasm. So as I was talking about cristae, so they are meant for increasing the physiologically active area of the inner membrane. Cristae, number one is increase the physiologically active area of inner membrane. Okay, and they are arranged generally like baffles at right angles to the longitudinal axis of the mitochondria. So if this is the longitudinal axis of the mitochondria, they are at right angles to it. This is the thing. Okay, anyway, and they are tubular. They are tubular in shape, and also a very important thing is the density of cristae. They indicate indicates the intensity of respiration. The second point, density of cristae indicates respiration intensity. Okay, so this is a very important terminology, that is respiration intensity. Okay, now the next thing is elementary particles. You see here are certain bulb-like structures which are protruding out, or rather inside the cristae only, but towards the inner membrane, towards the inner chamber. And these elementary particles are also known as oxysomes. Here is a magnified structure of oxysome. It has a head, it has a stalk, and it has a base, and their dimensions are already mentioned. So these are small tennis racket like particles, and they are also known as F0, F1 particles. And F1 is both the head and stalk combined, and F0 is the base. So these elementary particles, they have channels which are meant for passage of protons. So protons, because we know that we have studied in the previous slide that they are less permeable, the inner membrane is less permeable, so these are certain passageways for protons, and they function as ATPs. That means they are an integral part of the ETS, the electron transport system. You will learn about this in much more details when we talk about ETS in respiration. So these are integral part of ETS, and they are providing passage for H plus, for protons. And enzymes of ETS, they are located in the inner membrane, contact with elementary particles. Okay. So ATP, how they are functioning as ATPs? Basically, during oxidative phosphorylation, they are a proton gradient is built up across the inner membrane. So the inner chamber becomes, that means the matrix becomes highly laden with H plus, and when these passageways are opened, they are releasing the tension. So according to their electrochemical gradient, the H plus flows, and using that energy, using that tendency of H plus or protons to move from one chamber to other, they're harnessing that electrochemical gradient energy into ATP, which is the chemical coin for energy, and likewise they are producing ATP, as simple as that. Okay, one more important thing is presence of adhesion sites. So at places the outer and inner mitochondrial membranes, they come in contact with each other, and those are known as adhesion sites. Adhesion sites. Adhesion sites are special permeation regions for the mitochondria for transfer of materials from outside to inside and vice versa. Okay, so the outer chamber, that would be the the perimitochondrial space. We have already talked about that. And inner chamber, the core of mitochondria. And in the inner chamber only we have all these things: the DNA and the ribosomes, 50S or 70S types, and but DNA can produce its own RNAs. So from there we come to the autonomy of mitochondria. I said that DNA can replicate itself independently and can also produce its own RNA, then mRNA, tRNA, and the ribosomes, which are 70S types, they resemble prokaryotes. The mitochondria can grow internally also, and new mitochondria develop by division or binary fission of pre-existing mitochondria. So all these points suggest that they can live independently, but then they're not completely autonomous. They're not fully autonomous, because there are certain proteins which are required by mitochondria, but they get translated due to the genetic material which is found in the DNA of inside the nucleus. So some part of it is also controlled from the nucleus. So that is why it is not entirely independent. But this points that they have their own DNA which can replicate itself, which can transcribe itself and convert into and form various RNAs, mRNAs, tRNAs, own ribosomes. These led to a proposal from Margulis in 1971, and that mentioned that these might be symbionts. These might be symbionts, and not exactly an integral part of cell from the very beginning of life. Rather, they came as parasites, and then they became one of eukaryotic cell organelles, and that happened through evolution. So this was first proposed by Margulis in 1971, that mitochondria, they are symbionts in the eukaryotic cells which became associated with them through evolution. So this is all about mitochondria for today. In next class we will continue with cell again. Till then, stay safe. Thank you guys.